In today's class, we are going to discuss about the process of protein synthesis second half, that is translation. As we all know, translation is the process by which we make the protein chain, that means the polypeptide chain, as per the codon sequence or the messages on the messenger RNA. Translation process is a complex, multi-stepped process which is taking place inside the cytoplasm and especially at the cellular level, that is in the organelle layer, ribosome. Now, when we look at these multi-stepped complex process of translation, we can see 5 different phases are there in the translation process. We will learn one by one. The first and the foremost step in translation is the amino acids activation. We are all knowing that the cellular level, the translation takes place by the help of the raw material amino acids. We have 20 amino acids scattered throughout the cytoplasm. So from the cytoplasm, suppose this is the cell and this is the nucleus, we have so many amino acids scattered throughout the cytoplasm, and 20 amino acids we have found out so far. These amino acids should be brought to the ribosome. For that, we require certain steps. The first step is the activation of the amino acid. We need to activate these amino acids. And how can we activate these amino acids? The ATP source is ready in the cytoplasm. So when ATP combines with the amino acid, we require a special enzyme. The name of the enzyme is amino acyl synthetase enzyme. So when the amino acid combines with ATP and with amino acyl synthetase enzyme, we get a complex of all the three. So this complex is amino acyl synthetase complex. Rather, in a brief we will write amino acid plus AMP. How come ATP turned to AMP? Whenever ATP is releasing energy, we can see that the high energy rich bonds are broken. So one by one ATP's phosphate groups are removed. So here 2 inorganic phosphates are removed. So naturally adenosine triphosphate will turn to adenosine monophosphate. So 2 phosphate groups when removed from ATP, adenosine tri turns to mono. So here the complex is named as amino acyl synthetase complex. And at this point of time, you can call this amino acid as a activated amino acid. Now we can see that the role of transfer RNA, because scattered amino acids must be now attached with the transfer RNA. Transfer RNA is normally clover leaf shaped or inverted L shaped. So we can call it, we can draw it in the form of these inverted L shape. So when we look at this transfer RNA, you can see that the same complex is combined with the transfer RNA. In our previous video, we have seen that at the 3' end of transfer RNA, the amino acid is getting attached. So here is the attachment. As a result, we can see that fully attached the amino acid with the transfer RNA. And this process is also called amino acylation. And here comes amino acid combined with the transfer RNA. Now we do not require the enzyme. So enzyme gets freed off, and at the same time AMP is also not needed. So both these components of this complex is getting free and we require only amino acid, that amino acid is combined at the 3' end of this transfer RNA. So the second step of charging of transfer RNA or amino acylation is over. Now we have to slowly come to the ribosome, the integral part of the translation. When we look at the ribosomes, we can see that in a normal condition, ribosome constitute two units, a big subunit and a small subunit. Small subunit is 30 Sedgwick unit and the large subunit is 50 Sedgwick subunit. But normally they don't attach together. They are freely as well as separated form. But when messenger RNA leaves the nucleoplasm, comes along with this small subunit, free small subunit, immediately this free small subunit will find out the large subunit. So at this point of time, you can see that both subunits are coming close to each other, and the figure is shown over here. And when we look at the large subunit, it has got two reactive sites, very essential reactive sites. The first reactive site is A site. A site is technically called amino acyl site. At this particular site, the transfer RNA is bringing the amino acid, and that is why this is called the amino acyl site or A site, which can ever receive the amino acid. There is a second site which we call as P site. P site stands for peptide bond formation site. Whenever the amino acids are coming in a sequential way, we have to immediately make the peptide bond. So that particular site is what we call P site. There is one more site which we can see, that is the E site. E site is technically called the exit site. Through this additional E site, the peptide chain is exiting out. Now we can see that how the elongation of the amino acid chain takes place. We are all knowing that one by one in a sequence, as per the codons on the messenger RNA, the amino acid will come. And this sequential chain of amino acid should be getting added one after the other. So that is what we call elongation of the peptide chain. This elongation of the peptide chain is mainly by the bond formation. We call this bond as peptide bond. So this peptide bond formation takes place at the P site. So you can see that this initiation is always by the help of a triplet code, that is AUG. AUG stands for the initiation codon, methionine. It stands for methionine amino acid, at the same time AUG. So AUG whenever comes on the messenger RNA, the step initiation begins. Once this AUG comes at the P site, slowly the elongation begins. We can see that UCU, serine. So you can see that serine is the amino acid coming here. This serine carrying amino acid will be read by the triplet codon of the messenger RNA. Suppose this is the serine. So serine comes at this site. So slowly this serine carrying transfer RNA will shift from A to P. So how this shifting of A to P will take place? Slowly this ribosome moves, proceed towards the 3' end of the messenger RNA. And now this site is free, open. So towards this A site now, leucine carrying transfer RNA is coming. Suppose this is the leucine carrying transfer RNA. This will come and occupy the A site. So from the A site now serine has gone to P site. So this A site is now occupied with leucine. So after that you can see that glycine. So glycine is the third amino acid coming. So now the glycine will come to the A site, and from A site serine will go to the P site. This is slowly and slowly going through this messenger RNA, just like that. Suppose this is the messenger RNA and this is the ribosome. Slowly from the 5' end, from the A site to P site, this ribosome will slowly move. At the same time, you can see that the chain of the amino acid will be proceeding. And we can see that the peptide chain formation takes place. And how long this will continue? This will continue till the termination codon is coming on this messenger RNA. Suppose here on the messenger RNA, the termination codon UAG comes. So when this ribosome progress towards this side of the 3' end and suddenly UAG comes, what does it indicate? We have to stop this elongation process. That is what we call termination. We are going to end the synthesis of the peptide chain formation. So here we come to the termination. Termination can be made by 3 distinct codons like UAA as per the dictionary, genetic dictionary, UAA, UAG and UGA. I have here preferred and written UAG. This UAG when comes at this particular end, when this A site recognize that, oh, it has come with this UAG, there is no amino acid to take up, because as per the genetic dictionary, these 3 codons are not referred for any amino acid, so naturally amino acid will not be there. So here gives the signal of this termination. And one more thing is there, for termination there are some proteins termed as RF factors. RF factors or releasing factors, RF releasing factors, specialized protein which will bind with these termination codon. As a result, we can see that the polypeptide chain formation will be stopped there. That results in the end of protein synthesis, or we call it as termination phase. So altogether we have seen 5 distinct phases are there in the translation. Activation of the scattered amino acid in the cytoplasm by ATP. Then comes charging with transfer RNA along with the activated amino acid, that is called amino acylation. Then the whole ribosome, the machinery is getting geared up and ready for the initiation. So this initiation gets started with AUG, the methionine codon. This AUG when comes at the P site, you have to see that never AUG comes to A site. It can directly come to the P site, and slowly this amino acids are getting added up at the A site by the help of the codons of the messenger RNA. We call it as elongation by the formation of peptide bond. And when the messenger RNA takes up with any of these 3 codon, we can get the signal of stoppage of the polypeptide chain, and this is what we call the termination of the polypeptide. I hope that the complex process of termination, the translation is understood, and thank you.